Review of Type 1 Diabetes Clinical Trials for Stem Cell Treatments by Group 1. Type 1 diabetes is an autoimmune disorder in which the body's own immune system attacks beta cells, which are vital for producing insulin. Insulin is a hormone that allows the body to process sugars from carbohydrates and store them for energy. Type 1 diabetes patients require insulin to be pumped into their body for, as stated by a 2017 Mayo Clinic article, poorly controlled diabetes can lead to a variety of serious health conditions, including heart disease, stroke, blindness, amputations, kidney disease, and nerve damage. Although the exact cause of this disease is unknown, it is of genetic origin and is not to be confused with type 2 diabetes, which is induced from lifestyle choices and is caused by insulin intolerance, often onset later in life. The United Kingdom's government's diabetes resource outlines how developed stem cell treatments have attempted to cure type 1 diabetes through islet cell transplants, which help repair the pancreas to produce more insulin. Although the body's immune system still attacks these cells, patients who receive this treatment ideally will be able to produce enough of their own insulin to process glucose normally, either with the help of mild immunosuppressants or none at all.
decide whether or not a clinical trial was legitimate, we looked at how well each trial met our criteria. The first part of the criteria was what the credentials of the lead researcher or author of the clinical looked like. If a lead had never been published or did not have a doctorate in an appropriate field, we considered them to be illegitimate. Next, we looked at the cost of the participants. A legitimate trial would not charge their volunteers and would instead offer to pay their travel costs. Then we looked at how well the trial followed FDA regulations. Even though a trial is posted on the clinical trials website, it may not be legitimate. We also checked out the sponsors of the trial and researched their background. We checked what phase the trials were in, and then we looked at the studies themselves. We looked for a detailed description of the study, past the related studies, criteria for the participants, and the results of the study. If the description is sparse or the results were never published, then the trial seems a little off. However, a trial without published results does not necessarily constitute illegitimacy. Perhaps the trial just did not end the way the researcher wanted.
There were 8 completed or recruiting trials relating to stem cells listed for type 1 diabetes within the United States, reported on clinicaltrials.gov. However, upon examination, only 4 of those were determined to actually use stem cells for treatment, and of those, only 3 were determined to be legitimate. Not enough information was found in one of the trials to make a judgment. While the main researcher for this treatment was well qualified, the company in charge of the treatment did not seem to be well established. We chose 2 trials that seemed to be the most legitimate, as there was more information regarding the specifics of those trials available.
Examining the medication Procamol for the treatment of recently diagnosed type 1 diabetes. While the listing for the trial stated that it was in its second phase of treatment, further research showed that it continued on into its third stage, which increased its legitimacy according to our standards. Additionally, the study utilized a large number of participants, 63 to be precise. This made its results much more statistically significant than the other trials, many of which had around 10 subjects. Mesoblast, the sponsoring company for this trial, is renowned for its regenerative medicine, which added credibility to the study. In addition, the Juvenile Diabetes Research Foundation is listed as a collaborator on the trial site. The Juvenile Diabetes Research Foundation is a force in the diabetes research world, and so having their name on the study lends it authority. The study was listed as both double-blind and randomized, meaning that its research method were designed to produce accurate data. It was also available in multiplications, which adds credibility. Participants had the option to go to any number of high-profile research institutions to participate, meaning that all of these sites had some measure of association with the study, even more proving its legitimacy.
The second trial we determined was legitimate was a stem cell educator therapy. This trial is FDA approved, although it is still in stage 1 and recruiting. Researchers estimate a total enrollment of 10 participants. Michelle Donato from Hackensack University is the lead researcher for this trial. She has her medical degree, certificate in medical oncology, and has been practicing medicine for over 20 years. These credentials make Dr. Donato a qualified individual to carry out a trial such as this. Hackensack University is also the sponsor of this clinical trial. No patient cost was found on clinicaltrials.gov, and no results have been posted for this trial, although it is reported that the first round of results for primary report are expected to be ready in June this year. The selection criteria for this trial is also very reasonable. Participants must be at least 18 years old and have no other coronary artery or autoimmune disease besides type 1 diabetes. No pregnant or breastfeeding mothers can participate either.
Treatment by a stem cell educator therapy begins with apheresis. This is when the patient's blood is drawn out and separated into its components. The lymphocytes, or white blood cells, are separated from the other components, the latter being returned to the patient's body. The lymphocytes are then run through an assembly called a stem cell educator, which has layers of cord blood stem cell cultures. The goal is that the cord blood stem cells will change the immune response of the lymphocytes, so they have a less damaging response to pancreatic blood beta cells. The lymphocytes are run through the stem cell educator several times, but cord blood stem cells themselves never enter the patient's body. Cord blood stem cells secrete signaling molecules which educate the lymphocytes with the goal of restoring more normal immune function. The stem cell educator machine was first developed by Dr. Yang Zhao at the Hackensack University Medical Center. He performed a trial for type 1 diabetes in 2012, which became a precursor for this current trial. His results were very promising for the future of type 1 diabetes patients, and this clinical trial is hoping to build on this success.
Here are our sources. Thanks for listening.